Hello there, Per Alstrom calling from Uppsala, Sweden. I'm really very sorry I can't be with you in person, but I'm grateful to the organizers for inviting me to give this plenary talk, this keynote talk. And I'll start by sharing my screen. There we go. So I'll be talking about the superfamily Sylvioidea and what we've learned about the relationships and evolution of this group in the last few decades. To start by an overview of the passerine tree of life, we've got the Acanthicity, which today comprises 2 extant species of New Zealand wrens, as sister to all the other passerine birds, which in turn comprise the Tyranni, the sub-oscines, and the Passeri, the oscines. So here is the sub-oscine tree, and the Acanthicity as sister to that. We jump over to the oscine part of the tree, and as you can see by the colored charts here, it all started in Australia, in the Australian region, by the red boxes, and all of the early divergences, all of the early branches in this tree occur in the Australasian region. Then we had dispersals to various parts of the world within this large group here, the corvid radiation. And if we move up the tree into this group here, we have one major dispersal event into Eurasia, and we have subsequent divergence, radiation of the Sylvioidea, as well as a number of other groups of various number of species, some large, some small. So we've got, for example, the Bombyciloidea, Elacuroidea, Muscigepoidea, Reguloidea, Certhioidea, and the large group of Passeroidea. And these have dispersed across the world and multiplied. But we will focus on the Sylvioidea today. And the first attempt to reconstruct the phylogeny of this group based on sequence data was published in 2006, when we used 1 mitochondrial and 1 nuclear intron to infer the relationships here. And we found 13 major primary lineages within this tree. And they are represented by the bars and the names, as well as the arrows here. And those clades were well supported, but the relationships among them was very poorly resolved, very poor resolution. But we suggested that 10 of these should be recognized at the family level. And this tree was actually quite a dramatic change from the previous classification. So if we look at the tree and the classification by Sibley Monroe, published in 1990, they placed all of the ones highlighted in blue here in the family Sylviidae. So as you can see, that is not a monophyletic group. A few years later, we expanded both the taxon sampling and the number of loci analyzed. And we concluded that there were actually 23 different families within the Sylvioidea as we defined it. Some years later, this phylogeny was published. So this is based on whole genome data on so-called ultra-conserved elements, UCEs. And it divides the family, the superfamily into 30 different families. And you can see the number of species in the boxes on the branches there. You can also see that all the ones highlighted in red were previously considered to be warblers. And it's quite obvious that warblers is not a natural group. So the warblers is instead a group that is that consists of species that have similar living conditions. So they are insectivorous and they have similar lifestyles, and therefore they have evolved similarities in appearance. If we look at one of those sister groups, the sister clade, the clade comprising the two sister families, Panaridae and Alaudidae, the single species family comprising the reedling, the bearded reedling, and the larks comprising 100 species. This is a relationship that was first documented by Per Eriksson and his research group in Stockholm. And when they published their results, it was really surprising. Everybody thought, what? How is that possible? And if you look at these in detail, you see that the bearded reedling was previously considered to be closely related to the parrotbills, the Paradoxornithidae, which, as you can see in the tree here, is in a very different position. Whereas nobody would have guessed that the the reedling is actually most closely related to larks of all species. But in fact, it's not the reedling that is weird. It's the larks that are weird. Because if you look at the entire group here, the entire Sylvioidea, it's only the larks and the swallows and martins, the Hirundinidae, that occur in open habitats. All the others are found in some sort of secluded habitats, such as forests, such as scrublands, wetland vegetation, etc. Whereas the larks are all in open habitats, and the swallows and martins, as you know, are feeding on their wings. They're flying around and feeding. So they're in very different habitats compared to the others. If we look at the larks here, we did a phylogeny comprising approximately 80% of the species, and we found that the traditional classification, which was based on morphology, was completely off, completely wrong. All the different colors here represent different genera. So for example, Miraphra is here in this dark green. You see in several different places. You've got Calandrellus here and here and here and here and here. So the names that are written here are the ones that were proposed either in this paper or in an early publication, also based on molecular data. So it's quite obvious that there has been a very complex morphological evolution within this group, with lots of convergences and lots of really striking divergences. And at the moment, we're working on a revised phylogeny, including all except one of the species of larks, and using multilocus as well as genomic data for that. And we decided to split up the genus Miraphra into 4 different genera to align better with the other genera in this family. So if we look at one of those clades, you can see that the Calandrella larks, as they were previously defined, is not a monophyletic group, because because some of them are actually more closely related to the horn larks, to the genus Eremophila. So the Calandrella sensu stricto are more closely related to horn larks, whereas the Alaudala, as they were resurrected, are more closely related to Chersophilus and Eremalauda. As you can see here, Alaudala and Chersophilus, Eremalauda, they form a clade together. And what's equally surprising is that the genus Chersophilus is sister to Eremolauda, because these species, they don't look anything like each other. They're completely different looking. And in fact, the Dupont's lark in the genus Chersophilus, the monospecific genus Chersophilus, is pretty similar to the Southern African longbill lark complex, which is in a completely different part of the phylogenetic tree. Likewise, the Arabian lark, the Eremolauda, is very similar morphologically to Stark's lark, also of Southern Africa, which is also in a completely different part of the tree. If you look at the melanocorypha larks, the clade down here, you will see that most of the species look like typical larks, they're brown and streaked, except for the male black lark, which is very different. It's all black. But the female black lark is very similar to the other melanocorypha larks. The Mongolian lark was assumed to be closely related to the white-winged lark, but our phylogeny showed that the white-winged lark is actually not a melanocorypha. It's an Alauda, so it's related to the skylarks. So again, we've got convergent evolution here. So lots of cases of convergence as well as strong divergence. So as you can see on this map here, the larks are distributed mainly in Africa and the southern parts of Asia and southwestern Europe. So the red color represents the largest number of species, and the blue color is a small number of species. And in tropical Africa, you've got up to 16 species occurring together in sympatry. If we look at one of those, if we look at one of those genera, we've got the genera Eremophila, the horn larks. That was previously considered to be two different species. The Hemming's horn lark here, Eremophila bilofa, and the horn lark, the common horn lark, the rest. And as this tree shows, there are some really deep divergences among those horn lark clades, and the Hemming's horn lark is nested within the common horn lark. So it seems reasonable, based on this, to recognize 4 different species rather than 2 within the horn lark complex, within the Eremophila complex. And as is also evident from this tree here, there has been a single Pleistocene colonization of North America and subsequently down to South America from Eastern Asia. We can look at another complex here, the lesser short-toothed sandlark complex. They're usually treated as 2 or 3, sometimes even 1 single species. As you can see here, they are super similar to each other, really, really closely looking. And we sampled across the entire range of this complex. We sampled DNA, vocalizations, morphology, ecological variables. And based on DNA, we found 4 really deeply diverged clades and 2 subclades with slightly less deep divergence. We found quite striking song differences between some of these groups. And I'll start by playing the top one, the Heine group. It's got a rather long, scratchy song strophe. I'll next play the Chilensis, which is down here. It's quite significantly different. And there are, in fact, differences between all those in vocalizations. So here's a summary of what we found in this study. 4 main clades, 4 primary lineages, All of those differ in multiple aspects. So they differ in mitochondrial DNA. The tree is not shown here, but there are deep divergences between all those. They are deeply divergent in multilocus analysis, a coalescent-based multilocus analysis program called STACY. Morphology differs as well, song differs, and one of them, the right-tailed clade, the sunlark clade, it also differs in bioclimate and habitat. If we look at these two clades, the Chilensis clade and the Leucophea clade, as we call them, you can see that there are differences also in mitochondrial DNA, which is not shown here, but there's deep divergence. There is also, quite surprisingly, clear differences in bioclimate, habitat, but less differences in other aspects. Within this clade here, the right-tailed clade, we've got two different subspecies, and they differ in mitochondrial DNA, quite a deep divergence, but no difference in the multilocus data, quite significant difference in bioclimate and habitat, but less difference in other aspects. And within this clade here, there's no difference in the multilocus data or in bioclimate habitat. Vocalizations, we're not sure of, we haven't got enough data, but some differences, but not so great in other aspects. So we concluded here that this complex is appropriately treated as 4 different species. The family Setidae, the bush warblers and allies, is a group that occurs in Asia, mostly, also in parts of Europe and North Africa. We'll look at that in more detail in a second. But this tree here revealed some really unexpected relationships. All the species whose name are in black here were placed in the genus Setia based on morphological characters. You see they look fairly similar to each other, but brown with fairly long tail, rather short in this case, but brownish upper parts, paler below, pale supercilium, all looking basically similar to each other. So it was a great surprise when we found that within this group, there is a number of other genera which are here represented by different colors. And nobody had ever guessed that all of these really colorful things, for example, or these really short-tailed ones, would be nested within the genus Setia. So we had to propose a new taxonomy here. And interestingly, though, even though the plumages are so different, we counted the number of tail feathers of those birds because we knew that Setia was pretty unique among passerines in having 10 tail feathers. And it turned out that all of these also have 10 tail feathers. And with respect to these little Theseas, they have maximum 10 tail feathers. It's actually really difficult to count the numbers because they're so short. So just look at this. This is 2 of the sister species in this phylogeny, which look really, really different from each other. The chestnut-headed Thesia, Oligura castani coronata, and the gray-sided bush warbler, Setia brunifrons. They don't really look very similar at all. Another unexpected finding is that the so-called Hemithesia neumani, the Neumann's warbler, which is endemic to the mountains of East Africa, is actually sister to the pale-footed bush warbler, which occurs in the Oriental region. And interestingly, the Setia seti, the Chettis warbler, has got a really big range, and it's the only one that's expanded into the Western Palearctic. And it's also, while doing that, it's changed its habitat compared to all the other species. It occurs in wetlands, whereas most of the others are in mountainous regions, in dense vegetation, in forest. So it's not only the appearances that have changed quite dramatically with various rates of divergence. So also the vocalizations have really varying rates of evolution. So if we look at this sister pair here, the gray-sided bush warbler and the chestnut-headed tesia, we'll just first listen to the gray-sided bush warbler. And then the tesia. So quite similar to each other, actually. I would say much more similar sounding than similar looking in this case. In contrast, if we look at these two species, which were previously considered to be a single species, their divergence time is much more recent than the two species at the bottom here. So Horonis bruniscens sounds like this. And Acatitzoides sounds like this. So at least to my ears, these are more divergent than the two species at the bottom, despite that they are much younger and they look very, very similar morphologically. The leaf warblers, the family Phylogeny, are mainly distributed across Eurasia. And as you can see on the map there, the largest number of species occurs in the mountains of the Himalayas and central China, so the Sino-Himalayan mountains, with rather few species in the Afrotropics and across the Pacific Islands. And this tree here, this phylogeny, it contains all of the species that are recognized today. And the number of species has actually increased by more than 50% since the mid-1980s. And what this phylogeny also revealed is that there are many cases of really strongly conserved plumage evolution, as well as some really divergent, some strongly divergent lineages, as well as some convergences. So if you look at the ones with red text here and here, they were previously placed in the genus Caesurcus, but as you can see, that's not a monophyletic group. This one down here, Xanthoschistus, was also at some point in time placed in Caesurcus. So here is just an example to show that to show different migratory patterns. We've got Phylogeny borealis, the Arctic warbler, which all of the populations from northern Scandinavia to Alaska, they migrate to Southeast Asia in winter. And in contrast, the willow warbler, Phylogeny trochilus, all of the populations with a similar distribution to borealis, they migrate to Africa in winter. So we zoom in on one of those clades. We look at the Phylogeny reguloides clade to start with. We've got here 4 different species, 3 of which were previously considered to be the same species. They are parapatric, but then we have 1 pair of sympatric, apparently reproductively isolated species. And they look very similar, they sound very similar. we've also got another group here in the Philospus intensio clade. Two of the species in this clade are very similar, both in morphology and in vocalizations, to these ones, but they are sympatric and they are reproductively isolated. And as you can see, there's a really deep divergence between those two clades. Within this clade here, you see there are some quite divergent looking things. So two of the species are very different looking compared to the others. This one was previously placed in the genus Acercus, as I mentioned before. With respect to the so-called Pacific Island clade, as you can see here, there is quite a lot of variation in plumage as well as in bare part colors. And there is also quite significant variation in vocalizations within this clade, unlike in this clade and this clade here. So one of the interesting groups within the leaf warbler group is the so-called golden spectacled warbler. This was treated as a single species with a wide distribution across Eurasia, across the southern parts of Eurasia. And it has two closely related species in the same group, the gray-cheeked warbler and the white spectacled warbler. So this was treated as a single species, and we studied this for many years, together with several other groups of people also working on this. And it was concluded that it's actually a complex of species, no fewer than 6 different species. 2 of them occur in the Himalayas, 4 of them occur in further east in China and neighboring parts of Asia. And they look extremely similar to each other, although there are actually some morphological differences that are consistent. So for example, this one's got 2 white outer tail feathers. This one's got 3 white outer tail feathers. The songs are quite different. And the one I played now was this one here. And now I'm going to play this one here, which has got quite a different song. So it's high-pitched, it's faster, and it's very often got those little trills. We also found that there was widespread symmetry with up to 4 species occurring on the same mountain, mainly segregated by elevation. And we also found that what was previously treated as a single species is actually not a monophyletic group. So as you can see here, the gray-cheeked and white spectacled warblers are actually more closely related to Tephrocephalus and Berkey than they are to those others in this complex. So the original golden spectacled warbler, Phylogeny berkey, or say Circus berkey as it was called, is not a monophyletic group. And the different species here are quite old. So although the age estimates differ among different analyses, it's clearly quite an old group of birds. So in a later study, we used genomic data to infer the phylogeny. And we found that in contrast to the previous study, that Soror is sister to Ormiensis, Whistleri, and Valentini. In the previous study, these 2 were sisters. However, in some data sets, mainly slightly smaller data sets, we actually found Ormiensis and Soror as sisters, but with fairly low support. These are maximum likelihood trees. We also tried to use a multi-species coalescent-based analysis program called SNAP. And when analyzing large number of data, so really a lot of DNA, we consistently found Soror to be sister to the others in this clade. Whereas some of the analyses using smaller amounts of data, smaller number of SNVs, found Soror and Ormeances as sisters with high support. So these asterisks here indicate a posterior probability of 1. So which is the true phylogeny here? We've got alternative hypotheses of relationships, and could it be that there could be ancient ingression that is affecting the tree? The coalescent-based analysis, the SNAP analysis, are supposed to correct for lineage sorting problems, but it doesn't take hybridization, introgression into account. So the first author of this study, Zhang, he did some really clever analysis to try to sort this out. And he formulated 3 different hypotheses here. According to H1, hypothesis 1, this is a phylogeny, and there's been introgression between Ormeances and Soror. According to H2, we've got a different topology, but there's been introgression between Ormeances and the most recent common ancestor of Whistler and Valentini. And if H1 is correct, analysis of loci showing the highest levels of introgression between Ormeances and Soror should result in this topology here. So if there's been a lot of introgression here between these two, the tree might look like this. But that was not the case in the following analysis. So what if H2 is correct? In that case, analysis of loci showing the highest levels of introgression between Ormeances and Whistleri plus Valentini, that should lead to this topology. And in contrast, conversely, analysis of loci least affected by introgression, they should lead to this topology. So if there's been very little introgression here, then we will have this tree. And that's exactly what was found. So this tree, these 2 trees, one maximum likelihood and one SNAP tree, are based on the loci that are least affected by introgression in this case. So there was also other evidence of introgression between Ormeances and Whistleri, which was found in other analysis. So is one called D-Suite and Phylonet analysis. And both these found evidence of introgression between Ormeances and the most recent common ancestor of Valentini and Whistleri. Moreover, when 50 kb sequence windows, non-overlapping windows, were analyzed separately, it turned out that more than 50% of the trees would reconstruct the phylogeny that we consider to be the wrong one, whereas only 1/3 of the trees actually inferred what we consider to be the correct phylogeny. Here's another group of warblers, the Arctic warbler complex. That was treated as a single species with a wide distribution. And we sampled vocalizations and DNA from across the range here. We found that there was a really deep divergence between the northern ones, Borealis and Kennicoti, and Examinandus and Xanthodrias. So divergence times ranging between approximately 2 and 2.5 million years between those. We also found consistent differences in songs as well as in calls between these groups. So Borealis and Kennicoti here, Examinandus here, and Xanthodrias here, all sounding different. So we concluded that this is not a single species, it's actually 3 different species. Morphologically, they're very similar to each other, actually. Here's another interesting example. The Phyloscopus affinis was previously treated as a single species, Tickle's warbler, with this distribution here. And it was considered to be closely related to Phyloscopus griseolus, the sulfur-bellied warbler. Based mainly on mitochondrial DNA, the population of Tickle's warbler from the eastern part of the Tibetan plateau was described as a new species, Phyloscopus oxysinensis. So as you can see here, the divergence between oxysinensis and affinis in mitochondrial DNA is almost the same as between either of those and griseolus. However, when looking at biometrics, plumage, and vocal traits, there was very little, if any, difference between oxysinensis and affinis, whereas griseolus was always well separated from the others. Also, looking at genome-wide data, here's a structure plot showing griseolus and the 2 others together when K was set to 2. When K at higher Ks, we would get admixture here between oxysinensis and affinis, and a PCA plot would show the same pattern, strong divergence along the PC1 between oxysinensis and affinis versus griseolus, whereas these ones appear to be more clinally distributed. And these ones here are from this area here, which is in between those 2 main distributions. And a tree based on a large number of nuclear imprints couldn't find any difference between these 2, affinis and oxysinensis, here just analyzing non-admixed individuals, so individuals from outside of this area here. And the conclusion here was that there has probably been an original species in this area, a proto-Tickle's warbler, and another species which is now extinct occurring in the Himalayas, and a shifting hybrid zone that has moved west. And while doing so, this species, this Himalayan species, has gone extinct, but it has left its mitochondrial DNA as a ghost lineage within this population. The babblers, they were previously treated as 3 different families, well, actually 2 different families. Well, babblers, classic babblers, and then white eyes, and then the Sylvioidea part that was left after lots of them had been removed from Sylvioidea. But now 7 different families are recognized here, and it's a large group of birds with lots of taxonomic changes that have taken place as a result of these molecular analyses. And you can see the ranges of the birds here in this. You can see that most of them are distributed in the Oriental and African parts of the range, and only the Sylvioidea are in the Palearctic, in the Western Palearctic up here. There's one species within the Paradoxornithidae which has actually moved across to the western part of North America. That's quite unexpected. But as you can see here on this tree here, all the bright green dots represent the Sino-Himalayan mountains. So this is an example of a group, or well, a big radiation with now several different families that have exploded out of the Sino-Himalayan mountains and got established in many different areas. And it was concluded that there's been a very slow decreasing speciation rate, except in the white-eye clade in the Zosteropeidae, where you see here really explosive speciation. That's this part of this figure here. And also interestingly, if you look at the number of species in relation to the time, you can see that there's a gradual accumulation of species. But in island species, you have a really sharp increase here. So a rather steep increase, and then even sharper down here. And that is correlated time-wise to these really strong sea level fluctuations. So it seems quite likely that the speciation rates have been connected to the changes in sea levels that have affected the appearance and disappearance of islands. We move over to the family Locustelli, the grassbirds and allies. As you can see on the map, they are distributed quite widely across the Palearctic oriental regions, all the way down to New Zealand and Australia, and also in large parts of tropical Africa and Madagascar. So the tree on the left here includes approximately 60% of the species, and the different colors represent different genera. So here's Bradipteris in red, here is Locustella in black, and the other colors represent other genera. So you can see that the phylogeny did not agree very well with the traditional morphology-based classification. So we proposed a revised taxonomy of these birds. And as you can see, there is quite a good agreement between geographical area and taxonomy in this case. Some years later, we published a study where we used the same number of loci, but 95% of the species. And as you can see, again, there is several cases of paraphyletic or polyphyletic genera, represented by the different colors here. So you got Megalurus in 4 different places here, for example. And it's quite obvious that there has been a lot of convergent evolution between Bradipteris and Locustella. And look at this funny bird here, the Malia grata. Single species in the genus Malia. It was actually found to be a Locustelli by Oliveras et al. A few years earlier. So that result was corroborated in this study. Here's another case of a bird that's diverged quite dramatically in appearance, but only the male in this case. The female is more similar to some of the other species. The genus Chenicola previously comprised 2 different species, this one in India and this one in Africa. And the phylogeny showed here that they are not sisters. Instead, the Indian one is sister to another Indian species, which was placed in the genus Chatornes, the single species genus Chatornes. So this is really surprising because these 2 are extremely similar to each other, and they've got some plumage traits that are pretty unique among all those birds. If you look at the nodes here, you can see that there is really, really poor support for the branching orders. And therefore, at least in theory, this one might be connected to this branch here. But in that case, the plumage similarity is due to a plesiomorphic state, so an ancestral state, and this one has diverged in plumage. More data needed to try to sort this out. There are other things in this phylogeny that needs to be studied further. For example, these two look very similar to each other, but one is down here and the other one is up here. So is that the case of convergent evolution, or could there be some other explanation? I honestly think that the last word's not been said here. I feel a bit skeptical of the position here, and as you can see, it's poorly supported, so it might be sitting somewhere else, perhaps as the sister to the others here. Again, more data needed to sort this out. But we had to revise the taxonomy once more, so this is what we came up with. The Cysticolidae were analyzed in a paper published some years back, and this phylogeny includes all of the genera in this family. And as you can see here, there is one clade that is restricted to Africa. There is one clade here, one or two species, which are restricted to the Philippines. This was actually found already by Oliveros et al. 1 year before this paper was published. And here we've got a more widespread clade, and also this one is slightly more widespread. This one is restricted to Madagascar. So let's look at one of the species complexes within this family, the Prinia crinigera, striated Prinia, and Prinia polycra, brown Prinia complex. So this was treated as two different species, the striated Prinia in this part of the range and the brown Prinia in this part of the range. And we found that they have rather different songs. So there are two different main types of song. One is restricted to this group, and the other one is restricted to this group and that one here. So this is what they sound like, just examples of these two main song types. So that was the striated Prinia type song and the brown Prinia type song, very different. It actually turned out that this is an ancestral song type which can occasionally be sung by these guys down here, but very rarely. So a multivariate analysis of vocalizations found quite strong differentiation between these three groups here. So Eastern birds, Western birds, and Central Myanmar birds. And in this group here, there was strong differentiation of Rocky in South and Central Vietnam, whereas these birds from other parts of Southeast Asia and Javan polycra were basically indistinguishable, so they sort of merge very much with each other. Based on molecular analysis, here's a tree based on mitochondrial DNA. You see that the striated Prinia is separated into two different groups here, so it's not monophyletic. Here's the so-called brown Prinias, and based on a multilocus analysis, coalescent-based program, it's again supported that the striated Prinia, striata, and crinigera do not form a clade. So taken all together, including studies of morphology, we concluded that this is actually 5 species instead of just 2 as previously thought. So here's the revised taxonomy of those. The spotted wren babbler has actually done quite a big leap in the tree of life. It's distributed in mountainous areas across Southeast Southern Asia. And it's got a really, really weird song. I don't know any other bird that sings similar to this. It's also weird in other aspects because it was considered to be a wren babbler. You've got an example of a wren babbler here. You've got the spotted elacura, as it's now called here. And the wren babblers are in the family Timalidae in Sylvioidea, whereas the elacura was found to be sitting on its own branch. And we propose that it should have its own family name, and it's actually so divergent that it should be placed in its own superfamily, Elacuroidea. As you can see here, there's poor support for the branching orders here, but the study by Oliveros et al. 2 years ago showed that it's actually sister to the Muscicapoidea. So very weird case here. So thanks very much for the attention and for giving me opportunity to talk about some of the research that I've been involved with. And I would really like to say thank you so much to all of my collaborators over the years, over the many years. And also, I wish to acknowledge people whose studies have not been cited here because I've mainly been focused on research that I've been involved with. But of course, there are lots of other studies that have been published. I wish to acknowledge my co-authors on the next slide, where you can see a list of the publications that were cited on those slides. And of course, I want to thank all the funding agencies that have made this research possible. So thank you very much for your attention and have a nice day.